bu beaucoup de verres de vin, je peux parler couramment le français. Mais malheureusement, lorsque j'essaie même de parler scientifiquement français, I become completely blocked. So I'm going to speak in English, and I hope you'll forgive me because I always admire so many of you who come along and speak English when it's not your mother tongue. Anyway, how do I get the first one? Oh, good. Okay, so can you hear me if I move around? Can everyone hear? Yes? Or do I have to stay here? Oh, I have to stay here. Okay. Right, I'll stay here. But, I have it on here. It's fine. I didn't see I had this. So I can give you an idea first of the outline of my talk, and then I'll go through each of the different parts. So the first part I want to talk about is the difference between domain general processes versus selective domain-specific processes. And I'm going to offer a third alternative, which is rather different, which I call domain-relevant processes that become, over time, once we reach adulthood, domain-specific. I then want to show you some examples of domain relevance in cross-syndrome comparisons. I'll then go on and say, but we shouldn't only look at genetic vulnerabilities. We have to look at how the environment changes for even normal children, but certainly for children with genetic disorders. And finally, because I was asked to do so, and it made me think a lot, I want to come up with some implications for training, for intervention. Okay, so to start, domain general approach is, of course, my mentor, Piaget. So Piaget had three domain general processes: assimilation, accommodation, and equilibration. And what was particular to Piagetian theory was that he applied these three processes to every domain of knowledge: number, social cognition, language, physics, and so forth. And while I think that's very important epistemologically, it didn't always help us psychologically. One of the things it didn't help us do was to account for very uneven profiles. How could a child be brilliant in mathematics and very poor in, say, language or physics? Why was that uneven profile possible? Of course, he talked about décalage, but those were décalage in timing. I'm talking about, even in the end product, uneven profiles. And this is particularly true in developmental disorders. Second, because of the big influence of Chomsky in the 60s, 70s, Fodor in the 80s, many developmental psychologists outside Geneva, at least, some inside as well, decided that the domain general view was not a correct one, and they opted for a domain-specific view. And we see that across many domains. I'll show it to you as an example in evolution, in neuropsychological patients as adults, and in developmental approaches. So this is my funny cartoon of evolution. And quite seriously, I had another picture that I couldn't find of Tubi and Cosmides holding a human brain, one of them, and holding a Swiss Army knife, the other, and saying that the human brain is like a Swiss Army knife. Every single part of it exquisitely fashioned for a particular purpose. So the idea was that evolution passed down isolated modules that were particularly suited to different purposes. My guess is that even with the Swiss Army knife, and I could be corrected on this, if you have one with 52 different features on it, you probably only use it about 8 or 10, and you use them for many purposes. You don't use each one for a single purpose. And that makes me think that maybe that image for the human brain is the wrong one. But it also occurred very much in adult neuropsychology. So you see here two cartoons, one of dyscalculia, so adults who have brain damage after their brain has been mature, and the rest of the system looks fine, but they can't do things in number. Or you might have grammar knocked out, so the patient will retain lots and lots of words but cannot string them together in a coherent grammatical way. And quite a few people applied this to developmental disorders. So they argued that there were developmental disorders like developmental dyscalculia, where the only thing wrong with the child was mathematics. Everything else looked, quotes, normal, and I shall come back to that. Or the child might have grammatical specific language impairment, and in that case, just the grammar was impaired and there were no other impairments. Again, these are exaggerated claims, but we'll stay with them for the moment. And what's wrong with this approach is that it doesn't distinguish between brains that are developed and mature and get damaged in their mature adult state and brains that are developing over time. I hope that's not a difficult thing for the translators, and thank them for all the efforts they're making, because I really want to make a distinction between the past tense in English developed and the idea of progressive, and you have it in some language, aspect in developing brains. So here's an approach. It's a quote from a colleague, Christine Temple, and her colleague Harold Klaasjen. And they took this very strict adult neuropsychological approach to a developmental disorder. And they said Williams syndrome can be explained in terms of selective deficits to an otherwise normal modular system. So the notion is you've got the brain, parts of the brain that are completely normal, completely intact, and just a few parts that are impaired. And that's my image of that as a cartoon to show you what is meant by this claim. And I will criticize this kind of approach very much during my talk. One of the main problems, and a problem that Piaget would have highlighted, is that these approaches ignore the developmental history of the organism. Organisms develop over time. Their brains develop, their cognition develops, their bodies develop as well. Reaching changes as your arms grow longer and longer, and so forth. So there's a developmental history that has to be taken into account. And for me, the adult neuropsychological model is therefore completely inappropriate for developmental syndromes. And if you think developmentally, then you might accept domain specificity of the adult brain, but your notion would be that it might not start out that way. It might become domain specific over developmental time. So not necessarily the starting point, but maybe the end point of a developmental process with its developmental trajectory. So in a lot of my work, and in the book that Jean-Paul mentioned, Beyond Modularity, I argued that the human brain didn't have built-in modules, as Fodor and Chomsky and a lot of developmentalists in America were claiming, but rather there was a progressive modularization. So very gradual, over time, specialization and localization of function. So if we reject the idea of domain-specific modules as a starting point for development, is our only other solution a domain-general one? And what I want to argue is that there is a third alternative, and it's something I've called domain-relevant processes. And the idea behind it is the following. Initially, many different parts of the cortex and even subcortical structures in both hemispheres compete to process incoming inputs. There's widespread activity across the infant brain. But certain parts of the brain are just somewhat more relevant, somewhat more appropriate for certain kinds of processing. Imagine that you're processing sequential input. Well, maybe parts of the brain that have denser neurons are more appropriate to that kind of processing, whereas other parts of the brain might be more appropriate to holistic processing. Those kind of differences in type of neuron, as I said, the density of neurons, the orientation of neurons, and so on and so forth. These would kickstart a system, but over time, with competition, everyone trying to process everything, certain parts specialize for different things. So you get specialization, but only over developmental time. And this we call the neuroconstructivist approach, obviously based on Piaget's ideas and adding the brain as one of the constraints that we look at. So I want to give you an example of this neuroconstructivist domain-relevant approach, and I'll do it through a gene. Because I think it's a very nice example, and because there was a lot of hype about this gene over time. So this is a family, AE family. And over generations, the blocks in black, both female and male, over generations, certain children had very, very impaired speech and language production, particularly in the productive, but also in comprehension. And for years, nobody really understood what was going on until Simon Fisher at Oxford and Tony Monaco at Oxford discovered that those family members who were affected by the speech and language disorder actually had a mutation on the FOXP2 gene. So a very tiny part, tiny gene, on chromosome 7 actually was impaired, mutated in these children, whereas other family members with much of the genome very similar did not have that impairment and had no speech and language impairment. The problem with discoveries like that is the way people jump on them. So Pinker, Gopnik, Mirna Gopnik and others started to think in terms of a gene specific to morphological markers. That was a paper in Science. Pinker called it the dawn of cognitive genetics, as if you could go from genes right to the cognitive level without taking into account everything between the two. And the papers, of course, reported it as a gene for human language discovered, and everybody got terribly excited. Well, very quickly, it was discovered that this gene was highly conserved over many, many different species. So in the bird, people looked at birdsong. That was already closer to human language, but you'll see how I'm going to argue that you don't even need to do that in animal models. What they found was that FOXP2 expression in the bird was much greater while the bird, the chick, was learning its song, and then reduced greatly once the bird could produce its song. In other words, FOXP2 expression differed over developmental time. Then people looked at the mouse. My colleague at the Institute of Child Health, Andy Kopp, was particularly involved in this. And here they found that FOXP2 expression over developmental time changed where it was expressed in the brain. So first it was the amount that was expressed. Now we're looking at actual regions where it was expressed. And the brain expressed FOXP2 very widely at first. But as the mouse became more and more active in its environment and explored the environment rapidly, FOXP2 became restricted to circuits that are involved in motor movement, so particularly the cerebellum. So there's a change over time in where and how much expression of FOXP2 existed. Then the researchers went back to the human case and found that actually in the KE family, the impairments were not limited to speech and language. In fact, there were many other deficits. So one was just making oral facial movements, nothing to do directly with language. I don't know if I can do this, but if I go, just copying that sequence was very easy for the normal 2- and 3-year-old and very difficult even for adolescents and adults with this FOXP2 mutation. They also had all sorts of problems with fine motor control. They had problems walking on uneven surfaces, so gross motor control. They even had problems in the perception and production of very simple movements. I don't know if I can do this, but probably. So I've changed the rhythm from 2, 2, 2 to 1, 2, 3. And people with this FOXP2 mutation found that very difficult to perceive and certainly extremely difficult to copy, whereas again, a very young child who doesn't have the mutation can do it easily. So we have a highly conserved gene across, even the snake has it, birds, I said, the mouse, a lot of studies now of chimpanzees and the human. And what it seems to be involved in is not a gene for human language, but it contributes to the skilled coordination of rapid movement sequences and their timing. And you can imagine how this might impact over time on some domains more than others. So FOXP2 is not domain-specific to language, as many people had claimed in the literature, but it's most domain-relevant to language, because in the human case, one of the most important aspects of our behavior is the rapid movement coordination and timing of our speech. Speech is used all the time in communication and so forth, and this is a very important part. If we look at child development in the speech case rather than language, we see children become more and more proficient at outputting faster and faster and getting their tongues around. I had this very nice picture, I think it's here, of this little baby make sounds from very early on. They're trying to get their mouths to move quickly around different parts of the speech system. So, if we talk in terms of domain relevance rather than domain specificity, it actually makes the researcher ask very different kinds of questions. So we don't just ask about oral communication, like in birdsong, for instance. We generate other hypotheses. So if it's rapid coordination of movement and their timing, then obviously sign language is another candidate. If you don't hear and you use sign language, you have rapid movement sequences in the manual mode. And that should be impaired in somebody who has deafness and the FOXP2 mutation. We might look at the playing of instruments. Piano, violin involves rapid movement sequences of the hands, again, and their timing. And we would generate hypotheses about impairments in those cases. It might even be true of rapid dance routines like the Scottish square sword dance and so forth. Maybe there's a dance in Switzerland that I've forgotten that I could have given as an example. But the point I'm making is we don't just look at language which we see impaired in these children. We go back with other hypotheses which are much broader about how the system develops and interacts over developmental time and see which are domain relevant in that case. But we also ask very different questions of animal models. So we don't ask what in the animal is most like human language. What we ask is what in this animal's repertoire requires the rapid coordination of sequences of movement, and then we make our animal model and test that. I now want to go on with a couple of examples from cross-syndrome comparisons, first of Down syndrome and Williams syndrome. I'll tell you a little about both. And then of Williams syndrome and autism spectrum disorders. And in the first case, I'm going to look at domain-relevant deficits in the number domain in infancy. So first, a quick word about the two syndromes. I'm sure many of you know a lot about them already. So Down syndrome, trisomy on chromosome 21. All the genes are normal, but there happens to be an extra copy of some 300 genes on that chromosome. So there's too much gene expression in this case. Williams syndrome, 28 genes deleted, piece of chromosome 7. I don't have time to go into the details now, but if you want to ask about it, I can tell you much more about it. And here we have too little gene expression. They only have one copy of those 28 genes. The other copy has been cut out. So here are two syndromes. They can be matched roughly on IQ, general measures, but they're very different otherwise. And there are two ways of interpreting data. So we looked at infant sensitivity to number, or to the numerosity of displays, and I'll give you some examples next. And under the domain-specific view, if I had been a nativist, I would have said, yippee, I've got a double dissociation between two syndromes. I can send it off to science immediately, or even nature. It'll get accepted. This is a big finding. But unfortunately, under the domain-relevant view, the neuroconstructivist view, we get a very, very different and much more complex picture of the same data. And just a word about that, I really feel that in today's state of the science, whatever the domain, physics, chemistry, genetics, psychology, we have to embrace complexity. It's not going to be simple. It's not going to be one-to-one mapping with genes or anything like that, or even one-to-one mapping between behavior and brain processes. It's going to be very complex, and we just have to accept that and take it on board. So first, the data. We first looked at small number discrimination. So we know quite a lot about what babies can do. This is typically developing babies. When they're discriminating between 2 objects and 3 objects. And this is an example of that. This is different screens. We show the baby, and this is just a very short version of it. We show the baby pairs of objects, so here and here. That goes away. We show them a screen showing another pair, another pair, and another pair until they get bored. And then we change the number on one side. And what we're looking for is whether or not the baby orients longer when there's a change of number. If the baby looks the same on both sides, then they haven't noticed the change. If they suddenly get a renewed interest because it's not boring anymore, we're not doing pairs anymore, then you can say they've discriminated 2 from 3. Typically, developing infants can do this at about 3 months, and some even have shown it earlier. I don't think showing things earlier is particularly interesting. What's interesting is the actual developmental progression. So we tested infants and toddlers with Williams syndrome and Down syndrome at a later age, so they should easily have been able to do it. And that was true of the Williams syndrome toddlers and infants. They looked like the typically developing controls. But with Down syndrome, there was no significant difference between 2 and 3 in their looking behavior. We replicated this with dots, so we show them 2 dots, 2 dots, 2 dots, and change to 3, or 3 dots, 3 dots, 3 dots, and change to 2, because that was what people were using much more in the literature. We wanted to be sure that we had replicated this. And again, the results were the same. So in this case, but not in the next case, the Down syndrome infants were worse than the Williams syndrome infants, and they were equivalent in IQ, and the Williams were the same as the typically developing. We then looked at large number discrimination. So this is more like magnitude. You don't count, you don't know how many are there, but you just sense there's more here than here. So a similar kind of thing, we showed them either 8 dots and we changed to 16, or 16 dots and we changed to 8. And typically developing infants take longer to be able to do this than the small number. They do this at about 6-7 months when it's a ratio of 1 to 2, and it's about 9-10 months when it's a ratio of 2 to 3. It's more difficult. Again, we tested a group of infants between 10 and 39 months, and this time it was the Down syndrome infants who looked exactly like the typically developing controls, and the Williams syndrome infants who found it difficult. So, a different pattern. So what we have so far, if I'm a nativist, is a double dissociation. In Williams syndrome, small exact number is intact, and I'm purposely using these terms, and large approximate number is impaired. And in Down syndrome, small exact number is impaired, and large approximate number is intact. So everything seems really clear. We have a double dissociation across syndromes. It's very exciting. We've shown that there are 2 separable numerical subsystems. They must be innately specified if I'm a nativist, because look, it's happening so early in infancy, and therefore genetically determined. So, in Williams syndrome, 1 or more of the deleted genes on chromosome 7 contribute to a domain-specific deficit in approximate number and spare the exact number system. And in Down syndrome, 1 or more of the extra genes on chromosome 21 contribute to a domain-specific deficit in exact number and spare the approximate number system. Everything seems very clear, but, and this is where I'm going to undo that interpretation and show you a very different way of looking at exactly the same data. But first, terms like double dissociation, spared, and preserved are very static concepts. If something is intact or preserved, nothing has happened. It's just remained the way it was endowed by evolution and birth. And these are borrowed from the adult neuropsychological literature, where after development something might be preserved or spared, but it has nothing to do with developmental syndromes, in my view. And I think it's absolutely crucial to trace any particular domain of knowledge back to much more basic level processes as early as one can in infancy. So domain-relevant processes. So one of the questions we asked was, how do infants with Down syndrome and Williams syndrome visually scan numerical displays? Maybe the differences in number processing in these 2 syndromes are not specific to the number domain, but much more due to general differences in visual attention, visual scanning patterns, things like that. And we did a series of experiments. They've all been published, so if anyone wants them, I can send them, and I don't have time to talk about them now. But what we found was that in Williams syndrome, there was a strong impairment in planning visual saccadic movements, just moving the eyes to follow something that has occurred. And they also had a very serious problem with disengaging visually. They had sticky fixation on something they were looking at. In Down syndrome, we found very much the opposite. They were very good at planning visual saccadic movements. We have a very nice study showing that, and they look very much like the typical controls. But they had a much more serious problem with sustained attention. They couldn't fixate and remain fixated. And we'll come back to those two differences later. So we developed the following hypothesis, and we used eye-tracking technology to test it, that maybe when you scan a large array of dots, for instance, what you need to do is rapidly move your eyes around the global entity, if you like. But when you're scanning small displays, what your brain needs to do is to focus on individual objects. And we felt maybe that would explain why the Down syndrome are good at large number and poor at small number because they can't sustain attention, and why the Williams syndrome were very good at small number but very poor at globally scanning an array. And this is exactly what we found, and here are some examples. So it's back to front for me. Here's an example of a child with Down syndrome looking at each of the different, so we're tracking where the eyes move and how long they stay on each of the objects. But look at the infant with Williams syndrome. That infant only looks at a tiny part of the display and often is off the display anyway, but very, very small movements of eye. We then looked also at 16. Here the Down syndrome infant doesn't look at everything because obviously there's a lot there and it disappears quite quickly. But they're sweeping the array and getting a sense of quantity. Whereas again, the Williams syndrome infant remains on very few items. Okay, so we can interpret these data, as I said, in two very different ways, either with a hypothesis that comes from nativism and domain specificity, or with a hypothesis that comes from domain relevance and development. So under the domain-specific view, I had a beautiful double dissociation. And I was tempted to send it in like that because it was very striking. But we finally rewrote the whole paper in the domain relevance view, and it's finally been accepted. It was a hard struggle because people don't like complicated stories. And it's coming out very shortly in PNAS. But under this view, we have a very different developmental story about how eye tracking, saccadic eye movements, sustained attention, etc., contribute over time to the results we get in the domain of number. So let's again take Williams syndrome this time and just focus on it a little more for a moment. So they're very impaired in planning basic eye movements, saccadic eye movement planning. So it's clear that we've shown that this basic deficit early in infancy contributes to the deficits in discriminating large numerical displays. And we might stop there. We've done our job, we've shown what happens in number and so forth. But with a neuroconstructivist view, we don't stop at number. We ask the next important question, and I think this is one that's rarely asked, is this deficit also domain relevant to other domains? Could we find, although it's very apparent in this domain, more subtle deficits, but where it's also apparent in other domains? But not only that, could we also explain proficient behavior? So if behavior that's successful, not only deficits, with the same explanation. Mainly in neurodevelopmental studies, the deficit is on one side and the success is on the other. And we said, can the same mechanism actually explain both? So I want to give you an example from Williams syndrome face processing. I haven't looked at the time at all. Am I fine? Yeah, okay. In Williams syndrome, faces are proficient. They're in the normal range on several face processing tasks. And some people have interpreted this as the ventral stream of the cortex is spared. Number, as I've just shown, is impaired in infants and very impaired in later development in adolescence and adulthood. And here people argued the dorsal stream is impaired, and that explains number. What we asked was, could it be that the saccadic eye movement planning deficits, which gave rise to the problems in number, might be domain relevant to face processing as well, so it would explain not only the deficits in number, but also the proficiencies in face processing. And so we went about it in the following way. Many, many labs throughout the world, Bellucci's lab in America, Vicari's lab in Italy, Mills and so forth, and my own lab, have shown that if you give people with Williams syndrome, older children, adolescents and adults, standardized face processing tasks like the Benton and the Rivermead. So people have to look at a series of faces, at one face, and find them in another series of faces. Or they're shown a series of faces, and then those faces are interleaved with ones they haven't seen, and they have to say whether or not they've seen them before. People with Williams syndrome score in the normal range for that. So it looks as if it's, quote, spared. But we asked about the brain. Even though the behavior is good, is the brain actually doing it in the same way? And could we actually go behind the behavior and find that the actual cognitive processes were different too? I'm going to give you an example from the brain. And what we did was we used this high-density event-related potential net. So it's little tiny sponges, if you like, which send nothing into the brain. They just measure the electrical activity coming out of the brain. And what we looked at were human faces, monkey faces, and cars. And we did so in order to compare and see whether there was a specialization for human faces in Williams syndrome and compare them to controls. Now, we didn't this time work on infants. We worked on older children and adults because we wanted the system to be very proficient. We worked on the children who, in fact, passed the standardized tasks and got scores in the normal range. And what we found was that if you look at the healthy controls here, these two lines, the red line is the human faces, the green line is monkey faces. They show what is called the N170. So this is the amplitude, a negative amplitude, which is very typical of the brain saying, this is a face. Okay? And there's a small difference in latency because we're better at human faces than monkey faces. But cars look very different. The brain processes cars, if we're developing typically, in very different ways. But if we look at the Williams data, they actually show no difference between human and monkey faces nor cars. They all have a similar brain signature. So over time, the brain hasn't specialized for human faces. It can do human faces well, they pass the test, but they're not doing it in the same way as typical controls. We then looked at upright and upside down faces. And in this case, in the normal case, you're slower and you make more errors when a face is upside down. We have specialized in upright faces. And so what you see in the normal case, keep looking on the wrong side, is for upright faces, strong lateralization to the right, and for inverted faces, upside down faces, bilateral processing. And there's a big statistical difference between the two. If we look at Williams syndrome, there's no statistical difference between upright and inverted faces. They don't show what is called the inversion effect. And that suggests to us that although they're doing well on faces, they're actually doing it the same way as if a face were upside down. In other words, they're picking out individual features and not the general configuration. When we look at a face, we do it configurally, very quickly. When they look at a face, they look at individual features. And if you question them afterwards on the standardized tasks, they'll often say to you, yes, indeed, I looked at the cheekbones or I looked at the chin or something like that. You ask a typically developing person, they say, well, I could see it was the same. They just know. They see it immediately. So we have, in Williams syndrome, very proficient behavior, but a failure to specialize in the terms of the neurocircuits and a failure to localize in terms of hemispheric specialization. So we have very good behavioral scores, but they're underpinned by different cognitive processes, they're much more featural, and different neural processes, a lack of specialization and localization of function. So Williams syndrome face processing is featural. They focus on local details of the face. But recall, in the infants, Williams' success on small number discrimination was also because they could focus on very local parts of the display. And their failure on large discrimination was because they focused on featural details and didn't focus on the overall whole. So what I want to argue is that this featural bias in Williams syndrome is domain relevant to several developing domains. Only we don't always see it obviously when we're measuring behavior. It's very obvious in some domains, but it's quite subtle in others because they find other routes to reaching success. And for many people, they stopped testing Williams syndrome face processing because they got scores in the normal range. But we were curious and said, maybe those scores are coming from different cognitive and different neural processes, and it turned out to be the case. What about other cross-syndrome studies? So we've compared Williams and Downs, and it's a very complex picture. It's not nearly clear, double dissociations, etc. But what about autism spectrum disorders and Williams syndrome? And for autism spectrum disorders, I'm just going to say autism in future, but think in terms of the broader spectrum. Surely these are opposites. They have very different cognitive and social profiles. So maybe this time we really find some clear double dissociations. So let's look again at a little bit of detail on the two disorders. In Williams syndrome, genetically what we know now is that there are probably multiple genes of small effect and different sets of genes that explain the difficulties in social interaction, the difficulties in communication and language, and the obsessive interest in special behaviors. In Williams syndrome, we have 28 genes that are deleted, and we're beginning to realize now that 4 are of major effect. And this we do by looking at partial deletion patients, which I don't have time to talk about today. What about phenotypically? Well, people with autism show aloofness. They don't like interacting. They dislike looking at faces, looking at eyes. They prefer to interact with objects than people if given choices. They will interact with people, but it's not a pleasurable thing for them. And their behavior, most of their behavior on spatial tasks is better than their behavior on communicative and language tasks. Williams syndrome looks like the opposite. They're extremely friendly. They're fascinated by eyes and faces. You can't get them to disengage from eyes and faces. They much prefer to interact with people than to explore objects. And their communication and language is significantly better than their spatial tasks. So in the end state, in older children, adolescents, and adults, these two syndromes look very different. But we started to explore them in infancy and toddlerhood. And there you can find many overlaps, and I think this is a very interesting route to take. So both Williams and autism have difficulties with rapid movement, eye movement planning. They have atypical gaze following. They don't follow a person's gaze to an interesting object. They have atypical pointing and triadic interaction, so they don't follow pointing in the way that typically developing children do. They have atypical face processing. It's featural in both cases. They have heightened reaction to sound. They're both very sensitive to sound. They develop obsessions. They're different, but they're very much, if you describe them at the level of obsessions, it's clearly in both syndromes. They also have atypical sleep patterns, and we've recently been doing a lot of work on the effects of memory consolidation through sleep, and both of them suffer from non-consolidation of learning during sleep. They have problems with attentional disengagement, so they fixate on various stimuli. And what I want to argue, without going into any further detail, is that we might have a much better hold on the type of impairment that they have by comparing the syndromes rather than the obvious comparison with normal controls. If you compare, so we've done a study of featural processing of autism and normal controls, and the autistic group looked very, very featural. If we then compare the autism group with a Down syndrome group or a Williams syndrome group, the featural analysis is not quite right. You have to go into more depth because they're differently featural processes. So the cross-syndrome comparison is actually giving rise to a much more subtle understanding. And we can ask many, many different questions, but one of the things that we're doing at the moment is we're looking again at babies, because I think that's where you have to really look. And with a group led by Mark Johnson, Tony Sharman, and others, which is called the British Autism SIB Study, BASIS, and I have to take a plane at 5 o'clock in the morning or something tomorrow to get to their meeting. We're looking at very early markers of autism. So it's a longitudinal study. We have babies who are at risk of autism because another child in their family has been diagnosed with autism. And of course, we know that it runs in families and there's a genetic risk. And we're looking at family questionnaires, lab assessments of all sorts of different experiments, eye tracking, preferential looking experiments like the ones I showed you for number, event-related potentials, looking at the brain. We also look at resting state electroencephalography in order to look at the gamma band activity, which is to do with binding of features, because we're interested in how that takes place early on. We take DNA samples, et cetera, et cetera. But one of the questions I came up with at the BASIS meetings was, well, how do we know if this is specific to autism? Say we find these signs really early in development. Maybe these children don't go on to develop autism. It's just because they have a vulnerable computational system. So 2 of my PhD students are now looking at cross-syndrome comparisons using exactly the same protocol as in the autism study, and asking whether these early markers, because they're finding early markers, differences in the brain and so forth, are actually specific to autism or more part of a general vulnerable computational system that's growing. So we're comparing Williams syndrome, Down syndrome, Fragile X syndrome, and also children who live in very poor socioeconomic circumstances. And we believe that this will allow us to ask many different types of questions. Which processes are syndrome-specific versus syndrome general? Which are modality-specific versus modality-general? Are the cognitive and neural processes different, even when the overt behavior looks exactly the same? And we follow these developmental trajectories over time, rather than just comparing with controls. How much more time do I have? Okay. I said at the beginning that when you're dealing with genetic disorders, it isn't only the genes that change, there are also changes in the environment. So one of the things that I mentioned just now is looking at children with low socioeconomic status, children who are growing up in poverty. And there's a huge amount of detail. I'm just going to give you one quick example to give you a sense of what goes on. But we're looking at the brain and processes in the brain, many parts of the brain. And what we've found so far in preliminary data is that the frontal areas of the brain, the part that's involved in planning, monitoring, and so forth, already looks different in these children who are growing up in impoverished situations. So they have no genetic mutation, but they are growing up with environmental stresses. Now, this is the first study, I think, to show region-specific differences, and that's an important finding because the frontal area is so important in development across the board in many areas. But it's also the fact that these effects of poverty on functional brain activity are found as early as 6 months, which is very worrying, but it also is hopeful in that we can intervene, and I'll come to that at the end. But what I wanted to ask was, if you've got a genetic disorder, and certainly the children who volunteer to come into the lab, they are heavily stimulated by their parents, they have a lot of food, they're not growing up in poverty, etc. Of course, we have some like that, but most of them have really no environmental stresses. Is it just the mutated genes that explain why they are developing atypically? And what we've found is that it's not just the genetic mutations, but also the environment. So once a parent is told, your child has syndrome X, whatever that is, it automatically changes the social and physical environment. I'm certainly not blaming parents on this, so please don't misinterpret this. It's just a natural part of the interaction that one has with one's child because of the knowledge that one has. It's a little bit like gender differences. You treat girls differently than boys, even if you think you're not. I want to just give you two very quick examples. The first is from my lab, and we've been doing some observations of parents when they first come into the lab. So they're sitting down, there are 2 or 3 research assistants chatting to them, and the baby starts to mouth an object that we've put on the floor for them to play with. When that happens, in a typical child, the mother just makes sure it's not dirty and not sharp. We've, of course, made sure of that ourselves, and lets them go on, because they explore it with the nerve endings in the mouth and learn a lot about it. With an atypical child, there's a tendency to take it away because of a fear of danger, a natural fear of danger. These children are more vulnerable. The same with crawling. Child crawls away if it's a typically developing, and the parent just makes sure they're okay. With an atypically developing child, they're very frequently on their feet and picking up the baby and stopping that exploration. So, very natural reactions. You've got a vulnerable child, you'll have more fear of safety, but of course it stops the child having a rich environment to explore. In Carolyn Mervis's lab, she looked at overgeneralization. So a child might call everything a cat, and the parent of a typically developing child will say, yeah, it's a cat, it's a bit like a cat, but actually it's a dog. So they're kind of relaxed about overgeneralization. She studied a group of atypically developing children, and the parents there, the child would say cat, and the parent would say, no, dog. And it's a natural fear that because the child has mental disability, they may never learn the term correctly. So they stop overgeneralization, but that stops categorization and so forth. So again, there's an encouragement for exemplar learning. So I think we need a much more dynamic view of what the environment is doing. Now, very quickly, because I haven't got much time, I might run over 5 minutes if you don't mind. I was asked by Fanny Sisban, whom I'd like to thank for looking after me so well before I came to Geneva and since I've arrived, to think about intervention implications that will come out of the approach that I've been taking. And we've just begun in very recent times to do some intervention, but we haven't really spelt out the principles, and I'd like to at least mention 5 of them. I think there are many others. So first, what stems from this is that intervention should be early, before the cognitive level deficits have had time to consolidate. You get in on lower level processes much earlier. Training shouldn't necessarily be in the domain of deficit. So you know that the child might have dyslexia because they come from a family where dyslexia has been prevalent, and we know it has a genetic basis. We don't wait to train reading. We train oral phonological distinctions because we know that those are a problem in mapping later, so we get in really early. We train in syndrome-specific ways because syndromes differ in how they are vulnerable in development. Fourth, there should be a very active behavior on the part of the child, not being just told to do something or having something repeated, but the child should actually be in control of the training. I'll give you a couple of examples. And lastly, we need to measure the effects of training at the neural level. Only measuring it at the behavioral level will not tell us whether there have been real changes. So a quick word on each one. Why intervene early, and why at basic level processes? Well, first we know, as I've pointed out, and there have been many studies by Johnson and others showing that initially the infant brain is pretty unspecified. It has these domain-relevant biases, but they can change over developmental time. So get in when they are young. And we also know that neuroplasticity is much greater in infancy. It remains throughout our lives, even at my old age. I still am slightly plastic in my development. We know that there are changes in the brain in aging adults. Also, cognitive skills like attention, discrimination, memory, and so forth are domain relevant to several domains. So if you train those skills, you're likely to have an impact on the developing system. And as I said earlier, intervention should be before it affects the cognitive level. So because we have late diagnosis, for the moment at least, of autism, dyslexia, and certain other disorders, getting in early at the very basic level might change the developmental trajectory over time. And this was just an example of how the brain changes over time. I don't have time to go into it. I don't think it's important. Also said that training should not necessarily be in the domain of deficit that we know about later from research. So the example was number in Williams and Down syndrome. So what I want to argue is that in Williams syndrome, we don't train number, but we train rapid movement sequences. That should lead them to more global processing, and that should affect later the large number processing and other areas. But in Down syndrome infants, we need to train sustained attention because they have problems staying on target. They move a lot around it. And that should lead to more detailed local processing and also to small number processing. So that might give effects over many different domains. So here's a couple of very quick examples. We train the baby sees a nice aquarium, and every time the baby's eyes, so we use eye tracker, every time the baby's eyes fixate on this particular fish, everything starts to move dynamically. As soon as they move their eyes away from the fish, it freezes. So the baby is in control and learns gradually over time. If I keep my attention sustained here, all these interesting things happen. The same with this one. The little butterfly is flying across. Now it's moving across space rather than around in the same space. Every time the baby fixates on that moving butterfly, things happen around and the butterfly continues to move. As soon as the baby's eyes shift off the butterfly freezes. So the baby is in control via the eye tracking device of its own learning. I said training should be syndrome specific, so not one solution for all syndromes. And here again, one example is in Williams syndrome and Down syndrome, the delay in language acquisition is roughly the same early on. They change afterwards. So in the first 4 years, we've measured this, the delay is similar. But what we found is that in Williams syndrome, it's in part due to visual attention problems, watching people name things, following eye movements, and following pointing. In Down syndrome, it's partly due to auditory discrimination. They have much more difficulty in auditory discrimination. So the same language delay, going backwards in time, should have different approaches, auditory or visual. The child should be an active participant. So I gave you an example for sustained attention, but here's another one. The two microphones are just there to represent two different sounds. What we do is we train the baby to hear a sound, say a low-pitched sound, and every time he hears that sound, the window on the left, on the left opens, and every time they hear another sound, a higher-pitched sound, for instance, the window on the right opens. And after a while, when the baby has been trained to do that, we then don't open the window until the baby makes the eye movement. So the baby anticipates that mapping and then gets reinforced forward. And we can do that with many different things. Watch the screen now, not me. This is a way of training babies on their own without telling them anything. So you don't tell a child you've got to look at the eyes. You just make them learn that. So this is what the baby sees. Roughly, I've done it on the person has looked that way. There's a reinforcement, marvelous sounds and a visual thing. The child gets reinforced. By contrast, if the child looks in the other direction, so looks over here, they get a blank screen. And again, because things are seen also in peripheral vision, the baby learns that there's something about the relationship between eye direction and the stimulus. And we then look to see whether that generalizes to other situations. But as I said, you must measure the effects at the neural level. And this is just one example of that. So in normal, developed, healthy control children, looking mutual gaze, looking at one, shows a different pattern to averted gaze. This is also true in children at risk of other disorders, but not autism. But in the autism case, those two lines are very similar. The brain doesn't treat averted and mutual gaze in different ways. And what we will do is, after training, we will then look to see if that changes. If it doesn't, we go back to the drawing board and try another form of training. But even if they were to be able to make that saccadic eye movement, we still have to look at the neural level and see whether those have become differentiated. So those are the 5 intervention principles that I drew out, and I'm sure there are many others. So what I'm suggesting, and I'll end on this, is that when we plan intervention, we plan it neuroconstructively. We think about development over time. We think about how low-level processes impact on higher ones. And I think the most important question for all development, whether it's typical or atypical, and it's one that I know Piaget asked very, very often, and one that influenced me from those Piagetian influences throughout my career, are what are the infancy domain-relevant mechanisms that give rise to later cognitive level outcomes? What is the developmental trajectory at both the cognitive and the neural levels? And just before I end, I'd like to thank, above all, the baby scientists up here. We always give them a nice bib when they leave. But above all, my many students, my postdocs, and my collaborators, for all the work that I've talked about today. Obviously, it's done in collaboration with many other people. I've had funding from many sources. I just got funding from the Wellcome Trust, which is great. I can carry on until I'm 90. And I'd like to just mention a book that I have edited with Emily Ferran there, a young colleague at the Institute of Education. And most of the contributors are my colleagues from this developmental period of mine. Thank you very much.
Merci à Annette Carmilo-Smith pour cet exposé dont vous aurez apprécié l'extrême clarté et précision. Et en outre, on ne peut qu'être impressionné par l'ampleur des travaux qui ont été relatés ici. Et nous avons une petite demi-heure pour le jeu des questions et de la discussion. Qui aimerait intervenir?
Et vous pouvez poser vos questions en français, but I'll respond in English because I just don't speak French science.
Où sont les micros? Ici.
Thank you for this fascinating presentation. I would have liked to ask you, what are the works that you are doing, this connection between neuronal control and so on for autistic syndrome for adult autists, because this is where the problems have been least studied yet, and your plasticity of the brain development over time is encouraging, because I'm sure you're going to find new things. Are there any projects also for adults? Autistic syndrome adults.
Yeah, you mean future projects that are good. So, should come to the microphone. The BASIS study in Britain is part of a huge international study. So it's going on in France. I don't know if there are Swiss collaborators. I don't think there are at the moment. The work is certainly going on in Switzerland on autism. In America, all over the world, because you need big numbers when you're looking, obviously, at the genetics, but also at the brain. You need large numbers. But what I think is interesting is that there's becoming, and I think this is true in psychology generally, much more focus on individual differences rather than group data. So a lot of us are focusing on how do we analyze the brain data, for instance, in terms of individual patterns, rather than pooling the data and looking at group patterns. And I think this is going to be particularly important. with respect to autism, the genetics are suggesting that there are going to be lots of subgroups, that we're calling autism something that actually cuts across many different subgroups. And I mentioned earlier that there's one genetic study by Angelica Ronald and colleagues. They looked at the genetics, not which genes, but how much of the variation was explained in terms of genetics for the three major symptoms of autism. So the communication, the social interaction, and the obsessive interests. And they found that different sets of genes explained or accounted for each one. That suggests that when you look at the broad phenotype, you know, fathers of autistic children, uncles and so forth, one finds that they're not autistic, but they have some of the characteristics. One can imagine that they have some of the genetic mutations, but not all of them. And you have to have all of them. And because we have a dynamic system, if you've got one or two, you find a way of compensating in life. But if you've got all three, it's a real debilitation. So that's going on throughout the world now, and people are pooling their data. I think it's going to be a big movement forward. Just one final word. I'm taking a long time, but I'm very interested in autism. There is some suggestion that one subgroup may actually be due to a few genes that have mutations in copy numbers. But it doesn't explain all of the children with autism by any means. So when you find that, you look at a whole group and it doesn't explain it. Sorry, I took a long time. Where? Okay.
Hello.
Hello.
I've seen yesterday on news that there would be like a protein that would be a good therapeutic. They claim that it would be a good therapeutic target for autism. It's called neuroligin-free. Have you heard about it?
I'm sorry, I didn't understand.
Okay.
Can you repeat your question?
En français. Il y a un groupe de scientifiques à Bâle qui font des recherches sur une protéine qui serait impliquée dans l'autisme. Et ils clament que cette protéine, en fait, serait une bonne cible thérapeutique. Ça s'appelle la neuroligin-free. Est-ce que vous avez entendu parler de cette protéine et
Oui.
votre avis?
On peut peut-être compléter, si vous avez entendu cette émission à la télévision il y a 3 jours, je pense, où cette protéine intervient sur une synapse dont on a vu une photo. Mais ce qui est intéressant, c'est que ces chercheurs ont dit qu'ils allaient, avec l'industrie pharmaceutique de Bâle ou une maison de l'industrie pharmaceutique, trouver le médicament qui permettrait de guérir l'autisme. Et je pense qu'on peut comparer un peu cette proposition avec ce que tu as présenté sur les éléments d'intervention, puisque tous les éléments d'intervention dont tu as fait part portent sur des démarches quelque part comportementales.
Oui, but I know about the work. I know about a lot of work in the chemistry, the biochemistry of autism. Now, of course, those interventions may play a role. But my guess is they'll only play a role for one subgroup, or maybe 2 subgroups of children or adults with autism. The second is that when you intervene at the biochemical level and at the genetic level, because of the complexity of the system, you don't know what else you're affecting. So it'll be a long way off. I mean, people, make claims about I'm going to cure autism. They have to spend years with animal models. They then do clinical trials with humans on a very small scale because the implications are great. I mean, in genetics, for instance, a single gene has multiple functions, and many functions are involved. One function is involved in many genes. So every time you intervene at that level, you have to be very careful about the cascading effects over time. But I think all of these things are hopeful. Gene therapy, I mean, there's no doubt about the fact. Can I just go on a bit? Because we've just got a grant to look at Down syndrome and Alzheimer's disease. So in people with Down syndrome, 100% of them get the plaques and tangles of Alzheimer's disease. That happens all the time. But only 50% go on to develop clinically Alzheimer's disease. And nobody knows why. What are the protective factors? What are the risk factors? Our aim, a very long-term aim, after I've died probably, will be to intervene genetically once we can understand at the genetic and cellular and behavioral and cognitive and neural levels. But those are long-term projects, and people who stand up when they haven't even started doing the biochemistry and say, I'm going to cure autism, I think pay a very bad service to parents with autism. Because as Jean-Paul said, we need to do other things while we're waiting for these cures. We have to intervene at a much more local level, at the cognitive level.
Okay. Thank you. Yes.
Where are you?
Yes, here.
I can't see where's here.
Thank you. Me here. Thank you.
Who's me here? Oh, there.
Yes,
Sorry.
I'm far. Okay, thank you for a nice
Not that
speech.
I can see your mouth, but when I know where it's coming from, it helps.
Okay, great. Thank you. Okay, thank you for a nice speech. I mean, you have told us about a gene interaction environment for different disorders, and even though sometimes there is no mutation, genetically talking, there might be an environmental impact on those disorders, okay? But don't you think that at this level there might be some kind of epigenetic mechanism, such as methylation, that can occur into those symptomatic affect?
Well, yes. I mean,
have you worked on that level already or not yet?
My own, personally not, but I have colleagues, and I interact with these colleagues, who are working a great deal on the epigenetics of, say, poverty and so forth. So we know from many animal studies that the expression of genes is influenced heavily by the environment. We know that, from Michael Meany's work, for instance. So I think in the long term, you're absolutely right. We will use that kind of notion for intervention. At the moment, we just don't have a clear enough picture. But I agree. I mean, many researchers, particularly Caspi, and I've forgotten the other person's name, who are working on conduct disorder and looking at the effects of epigenetics on that. When you come back in, but my belief is you're going to have to come back in time and intervene very early. That could be wrong. It could be possible that we can do it later in development easily. My hunch is that will be not the case, that it'll be much easier and more effective if you intervene earlier.
Okay, thank you.
Thanks a lot for the speech. I was dumbfounded by your data about the difference in frontal activity in low-income and high-income families. And of course, the question that pops up at that moment is, well, have you got any clue as to what's the mechanism involved that creates the difference?
We don't know yet, no. I'm afraid to disappoint you and say no. We think it's probably in the frontal area because that plays such a wide role in many different things. So it's all to do with sort of planning and attention and so forth, whereas, say, parietal areas, temporal areas are much more local, if you like. But we were as surprised as you are that it was already there at 6 months. We took a gamble and we found it. Now, one of the interesting things is that the children at risk of autism also look, but this is still very early days, as if they show a similar pattern. So it could be that this is a particularly vulnerable area when you have anything going wrong, be it to the environment or your genes. The frontal area is... because we used to think that the frontal area only so-called came online late, but now we know that in very young babies, it's partially active very early. It's not fully active. So maybe that development that has to take place is vulnerable, and that's why you see it in these children. But we were surprised like you.
Okay.
Yeah.
Thank you.
You use a tool of brain electric answer, and I was thinking about the functional magnetic resonance imaging, and you a little bit speak about this.
Yes.
What will be the use in the future, because it's developing a lot, and also you show us the same good behavior by two different child, but with a bad neural functionment and a good one. Did you see different parts of the brain reacting for the face recognition, for example?
Thank you for that question. I had a whole slide on methodology, but it had to come out because of timing. We've used event-related potentials because babies allow it very easily. Our atypical children who hate things on their head, so we've got a big group of Fragile X children we're studying at the moment. They hate things on their head, they take that. We play with them with bubbles and things and just slip it on the head, and it's very light and it's good. Mark Johnson has one group of typically developing babies in the fMRI, so functional magnetic resonance imaging. You have to sedate them, so they have to be asleep. And ethically, it's quite hard to decide to sedate children with developmental disorders. We just can't get permission to do it. There's also the problem of sound, so you have to attenuate the sound a great deal. So what he is doing, and one of my students, we're co-supervising a student, is using functional near-infrared spectroscopy. And that's much less invasive. You know, it's also like a cap on your head, but it's measuring light, the function of light through the skull. And that's showing very interesting results. But I think the point of your question, I think it's a really important one, is we need different methodologies to show similar results, without which it could be spurious in one of those methodologies. Technology is improving by the day. I mean, the eye tracker has come in really recently. It was a big thing in the lab on the baby's head. Now nothing is on the baby's head. It just calibrates from the eyes. I hope in 3 weeks' time it'll be portable, because we have to take it into homes and we can't bring all our babies into the lab. The things are moving at a very rapid pace. Somebody is also trying to coordinate at the same time fMRI and ERP. We've done it with adults, but nobody's tried it yet with children. But I think from Mark Johnson's lab, that will come out soon as well. But the conglomeration of several methods, I think, is absolutely critical.
Oui, merci de votre intervention. Concernant l'autisme et la maladie d'Alzheimer, certaines pistes mettent en parallèle des problèmes liés aux troubles alimentaires ou à la toxicité. J'aimerais savoir, lorsque vous effectuez des tests, si vous prenez en compte ces facteurs ou pas par rapport au fait que chaque enfant, chaque jour, mange quelque chose de différent, ou s'il est allaité, qu'effectivement sa mère a mangé quelque chose de différent. Je vous remercie.
There have been so many different hypotheses about what causes autism. I'm sure you know about the MMR injection. That was shown, and lots of people stopped giving their babies injections against measles and whooping cough, and now measles is now epidemic again. It was shown to be completely false. Now, yes, I'm sure diet plays a role. But as I said really early in my talk, we have to embrace complexity. It's not going to be the only reason. None of these things are. The genes alone won't do it, because we have people with exactly the same genetic mutations. One gets sick, the other doesn't. We have identical twins. Their brains look different after they've experienced the world, yet they started off with identical genes. So I think you're right. We should think about diet. We should think about smoking. We know about fetal alcohol syndrome, for instance, so we're sure that alcohol plays a role when it's very heavy. But even then, people drinking exactly the same amount of alcohol, one baby gets fetal alcohol syndrome, the other one doesn't. There are so many factors that play. We have to embrace it, and we have to find what we do need to find. And that's something that I think is people are focusing on more, and I don't have the talent or expertise to do it, is models that allow us to analyze these very complex interacting factors. And then we can bring in rightly, a suggestion about diet, a suggestion about alcohol, all sorts of things like that, plus the genes, the cells, and all of the rest. Dynamic systems begin to address that. Bayesian analysis is helping, but nothing's quite right yet. But whatever it'll be very complex. Ouais. Il y a quelqu'un d'autre.
Bonjour madame.
Bonjour monsieur.
Je vous remercie d'être venu à Genève à l'auditoire Jean Piaget faire cette magnifique conférence. J'aimerais simplement poser une question, peut-être que vous pouvez m'aider. J'ai hérité du gène de la bonne humeur, du gène de l'intelligence, de la richesse, de la sagesse, et je suis à la recherche du gène de l'amour. Est-ce que vous avez une solution ou une proposition? Je vous remercie beaucoup.
I suppose I ought to reply, I'll meet you in a bar afterwards and tell you about the gene for love. I just hope that nobody ever says a gene for X. It just isn't like that. It's multiple genes and how they interact with the environment. But maybe love will be the exception.
Il y avait encore une question, peut-être la dernière.
C'est où? Ah oui.
Désolée. C'est une hypothèse. Vous avez dit que le cerveau se développait durant toute la vie. Alors, est-ce qu'on pourrait considérer que les maladies neurodégénératives sont aussi comme des maladies neurodéveloppementales?
Oh, ok, ok. Oui.
Même en français, ça marche pas.
Non. Si Jean-Paul a de la peine, I have an excuse. I think what's interesting is that over development and over evolution, certain syndromes have not disappeared. And you would think that if evolution just wanted the normal trajectory, they would have been cut out over time. They haven't been. So I think it's worth thinking, and I haven't really thought about this, I'm just talking off the cuff, about what it is that the differences bring to us. So some of them are just by chance, but some are inherited and so forth. Why didn't those particular things disappear? So there may be something about evolution that requires always difference, and that if we were to all move towards homogeneity, we actually would stop developing, that actually it is the differences. Now, I mean, I've often thought about, there are syndromes like autism, like Tourette's syndrome, where in some cases they're better than normal controls at certain things. So let's take an example from Tourette's syndrome. These are people who can't stop themselves swearing and having very bad tics. During adolescence, they usually have normal intelligence, so they're in normal schools, but they're embarrassed. You know, you're a guy, you want to meet girls, and you come out with all sorts of rude words, the girls don't like you. So they teach themselves control. It's very hard, but they do it again and again and again, every single day. If you measure executive function in these children, they're better than normal controls at the same age and same. So it suggests that there are some things that go wrong that actually enhance development. With autism, it's featural processing. They're brilliant at things like embedded figures, which we're really hopeless at. They're very good at it because they don't see the whole, they just see the parts. So there are developments that are actually positive. But that's not much of an example. I haven't really thought about it, but I think your question is a really important one. And we should think, you know, what has happened in the history of the development. Good.
Bien. Je crois que même si un petit rendez-vous qui est prévu, nous pouvons mettre un terme à cette séance en remerciant encore une fois très vivement Annette Karmilov de son exposé et de ses réactions. Bravo.